So go for the interview part. That's great. Happy to be here. All right.
Wonderful. Yeah, thank you. And so usually the question we ask is, what motivated you to become a scientist, or how did it happen? Was it something like a childhood dream, or it was something that came like an experience in life, or that
Yeah,
that
yeah,
that
yeah. It was a childhood dream, actually. I read um, The Golden Helix, Jim Watson's book about the discovery of DNA when I was a child. And, I had some other nerdy friends. So instead of playing cowboys and Indians, we were playing like Watson and Crick, basically, when we were kids. We were dreaming of, being like them, discovering cool things, and so on. And then when I was in high school, I grew up in Romania and Eastern Europe. And when I was in high school, I was in this high school that was sort of a special high school, a lot of mathematics, physics. I thought I was going to be a theoretical physicist because I wanted to figure out things and so on. And it dawned upon me that the human body is the most interesting object one could study, the most fascinating, the one that's sort of the internal ultimate frontier. So I decided to go to med school instead of going to study physics. And after I finished med school, I wanted to become better trained as a researcher. So I moved first to Switzerland and then to California at Scripps, where I did my graduate work in cancer molecular genetics. Then I went back and finished my clinical training at UC San Diego, and I wanted to bring some of those things that we knew in cancer to the study of the brain. Cancer at that time, which was 25 years ago, was not a mystery anymore. There were still a lot of details to be figured out, but we understood how, oncogenes, tumor suppressor genes, what made, cancer happen and so on. Whereas the mind was still something that was, a lot of blank territory, something that one could study for the next few decades and make an impact. So decided to focus on the brain, got some training in neurobiology, did my clinical training in psychiatry, and then started a career as a physician scientist in, studying the brain. And brought a lot of those things that were being used in cancer in terms of genomics, the idea of using molecular approaches to precisely assess patients, match them to treatments, try to bring that into psychiatry. And it's been a journey. It took 2 decades for us to develop this liquid biopsy approach for mental health that we're now, able to use actually with patients that can assess for various mental health diseases and dimensions. The most recent study was on anxiety, like the study that came out last week that you were mentioning, Katerina. And, the reason why it took so long and it's been so arduous was, on the one hand, you can't biopsy the target organ in our case, right? So unlike in cancer where you can biopsy a tumor and then match that against the peripheral profile, the liquid biopsy profile. In the case of the brain, you can't biopsy the brain in live individuals. So we had to rely on a very careful, stepwise approach to discover markers, candidate biomarkers to in the periphery, in the blood, to then prioritize them, to then validate them in independent cohorts of very clinically severe individuals, and then finally to test them in additional clinical cohorts for clinical validity to see if we can predict who's ill and who will have future disease exacerbation, hospitalizations, and so on. So we managed to do it. Otherwise, we wouldn't be having this conversation. It hasn't been easy. It's been a journey, but I think these types of approaches will make life much better for patients, will make much life easier for doctors, and will bring psychiatry into the 21st century to be on par with other medical specialties. And there's such a need for it. I'm also a practicing psychiatrist. I see patients, and the current way of doing things is sucks basically. Yeah, so you're sort of relying on what patient can recall, their subjective sort of perception, self-report on your perception of what they have. It's very important to bring quantitative tools to assess how they feel, how they think, to get lab tests, to put everything together to match them to the right treatment. So that's what we did, and that's where we're at with things.
Yeah, I think that's really wonderful. It's a wonderful life story, but and also the that you had the thought of why can't we have the same kind of precision from cancer for mental health. I think there are still a lot of misconceptions in a lot of people that the mind is something kind of I don't know, metaphysical, not something you can pinpoint with blood tests or anything. I think, do you think that this type of misconception was hindering the field for a long time?
Well, I
When
mean, not only sort of it, our approach can demystify things, but it also removes a lot of the stigma and fear and uncertainty that patients have. And I think just showing that, it's another biological disorder. There are biological abnormalities that you can identify, treat, monitor for, is very helpful to the patients. A lot of them tell me, I'm so relieved. It's not just something, that I'm imagining, and it's not just a weakness. There's something there, just like in diabetes, that can be detected and that we can treat for in a more precise way, and we can monitor for how well the treatment is working and prevent future relapses. So it makes things, as you say, it demystifies them, but also reduces this stigma and uncertainty that patients are facing.
Yes, it's so important. So thank you for that, pre-introduction. And yeah, the stage is yours. Everyone, the slides are pinned on top of the room. Feel free to access them. And yeah, thank you so much.
Yeah, yeah. So the first slide actually shows sort of the practical outcome of all this work. We managed to get some of these findings out of the academic environment, out of the ivory tower, and into a form that's usable by doctors and patients by doing a startup company called Mindex Sciences. Second slide, please. So we started, as I mentioned, on this journey 20 years ago. We didn't know it was going to be this hard and take this long, but that's what you have to do to make sure that what you discovered is real, that it's reproducible, that it has predictive ability, clinical utility, and so on. And next slide, please. So my research groups, my academic research groups, we named our laboratory from the beginning neurophenomics as opposed to neurogenomics. We wanted to pay attention to the phenotype as well, to phenomics, not just to the genomics part of things. I'm a geneticist by training, and obviously that's important, but being very careful what kind of animal models you use and what kind of ways of assessing people you use as part of your genomic study is as important as the genomic work that you do downstream of that. So we took a translational approach from the beginning. We did human studies, we did animal model studies, we integrated them, and that led to our ability to progress and to understand things better, develop some things that are practically useful, and try to educate the field about how to do things in a more precise way. Next slide, please. So when we started on this journey, psychiatry is sort of codified in terms of diagnosis in something called the DSM, the Diagnostic and Statistical Manual, and that's something that's like a phone book basically, and has hundreds of diagnoses inside and so on. The reality is a little bit, at least in my mind, is more simple. You have these. Are you guys seeing the slide with the Venn diagrams?
Yep.
Yeah,
Yeah.
okay. Yeah. So you have these three broad domains, the anxiety domain, the mood domain, the cognitive domain, and stress is a major trigger of these disorders. So over the years, we set out to systematically map the genomic landscape of these disorders in an empirical, non-hypothesis driven fashion so that we can understand the territory and then use that in a practical way. Next slide, please. So over the years, we've published studies on all these domains. We've mapped the territory. We need to add here on the slide the recent study that came out last week on anxiety. Basically, it was a sort of a very systematic approach from the beginning to map the genomic landscape of these domains. And stress and pain are triggers of the compensation in terms of mental health. You get worse anxiety with depression, sometimes dissociation and psychosis when you're under stress or under severe pain. We've looked also at alcoholism, which is a very common, addiction, and the fact that it overlaps those three domains is, actually an empirical accurate description. Some of the genes involved in alcoholism also are involved in anxiety disorders, some in mood disorders, and some in schizophrenia. So you know, uh, as I sometimes say, some people drink to be calm, so that's the overlap with anxiety. Some people drink to be happy, that's the overlap with depression, and some people need to be drink to be drunk to dissociate, and that's the overlap with schizophrenia. And we all have seen or have friends, colleagues, family members who sometimes succumb to one or the other of these routes. And next slide, please. So in order to do this, I moved from California to Indiana in 2004. Got recruited here as a young assistant professor, and I had a lot of funding for long-term genomic studies. It's a great place to work, nice people, very well trained, and also a very stable population. So we wanted to build a kind of a population study that has people that are followed over time, tested in the lab or in the hospital when they get hospitalized, blood samples collected, neuropsychological testing done, electronic medical records, and doing sort of genomics on them as well as phenomics, developing new instruments to assess them, apps, etc. So we started this cohort almost 20 years ago now, and at this point, we have over 1,200 actually enrolled in it, over 500 people that have over 1,200 testing visits in it. We've sequenced more recently our over 1,000 of their genomes. So it's been a very productive and very rich sample, but it takes work. It's sort of like a mini Framingham project, like that project that they had for cardiovascular diseases for decades in New England, where they followed people over years to derive insights about risk factors for cardiovascular disorders, cholesterol, and so on. We wanted to do something similar for mental health. So we put in the time and the effort long term to follow people over time, get multiple data points on them, multiple visits, and so on. Next slide, please. So we were able to, I think, to be successful because we had 6 innovations or things that we brought forward that were useful, turned out to be very useful. So number one, I already mentioned, learn from cancer, don't reinvent the wheel. So cancer has done a lot of work in terms of genomics, in terms of precision medicine. So over the years, we tried to adopt some of the same approaches and platforms. And as I mentioned, that was my previous field. I did my graduate work at Scripps in cancer molecular genetics. The second thing was this concept of convergence that we developed. We relied on convergence of multiple lines of evidence for identifying genes, biomarkers for pursuing them. We didn't rely on a single approach. So we had convergence between animal and human studies, convergence between multiple independent studies, and so on. So we call that approach convergent functional genomics. We, and I'll discuss it a bit more later. We also, um, we're very point out, the third innovation was phenomics. We were very quantitative and deliberate about how we assess phenotype. We developed our own visual analog scales to measure how people feel and think at that moment in time. Didn't rely on the classical questionnaires available in psychiatry, which are a bit of a hodgepodge of things. Number four is we were highly translationals. We did, a variety of translations from bench to bedside, from blood to brain, animals to humans, and so on. Number five is we took this very deliberate four-step approach to make sure that we discover, replicate, validate, test biomarkers. We didn't rely just on a single step and p-values, statistical significance, etc. We wanted replication as the key gold standard. And then in the end, you'll see how that all comes together into what we call the three circles of mental health. Next slide, please. So, when we started on this journey, this was a cover in Science. Cancer treatment gets personal. And that was, 2006. Cancer was way ahead of psychiatry, and we were just getting started to try to develop similar approaches. I think now, after years of hard work, we can say that psychiatric treatment can get personal. So, as a field, we've made it there. We caught up. Next slide, please. And in terms of phenomics, we remember those three Venn diagrams where you have anxiety, mood, and cognition. We think most people should be all people should actually be assessed and followed in a multidimensional space, not try to collapse them into a single dimension and say you have an anxiety disorder, you have a mood disorder, you have a schizophrenia. Most people have actually, function and have abnormalities on all three dimensions. So right now at this moment in time, if we assess for each of you your anxiety, your mood, your cognition, we can plot you in this three-dimensional mental landscape, mindscape as we call it, with your XYZ coordinate on these three dimensions. And over time, if we repeated those measures, you would be a cloud, your own personal cloud. And I think that's sort of a better way to describe people than trying to fit them into one dimension and into a DSM label. And most patients in clinical practice actually carry multiple diagnoses, which sort of reflects this fact that the doctor intuitively senses that they have multiple things going on with them. They just don't have the vision and the nomenclature to describe all those things. So then they stick multiple label on a patient, multiple diagnostic labels. Next slide, please. So, most patients are like a Rubik cube, a little bit scrambled and three-dimensional. And we think that with some of these precision tools that we're bringing to the table, we'll be able to have them all sorted out. So that's a cartoon from the New Yorker. All right. So blood biomarkers, which is what we're talking about today, are something that's very usable, practical in psychiatry. They are easily accessible. It's just a blood draw, as opposed to brain imaging, which is more expensive, more difficult to do. Can't do it sort of in any clinic anywhere in the field. You have to go be in a specialized imaging center, or even cerebrospinal fluid, spinal taps. Patients aren't super enthusiastic about getting a spinal tap, whereas a blood draw can be done in any setting, primary care, and so on. And if you have good blood tests, markers that can be used to assess and monitor people, then you're basically being able to deploy this everywhere. And that's the goal, to make this part of routine care, routine primary care, not just specialized high-end care in academic centers. So we wanted from the beginning to see if we can discover blood biomarkers that could be used anywhere. The So why would there be biomarkers in the blood that track or reflect what's happening in the brain? Well, one of the reasons is that the nervous and immune system are very interconnected during development, and also uh, afterwards, all the way to adulthood, all the way to through your lifespan. There are strong bidirectional brain immune interactions. The function of the brain affects the immune system. The immune system, cytokines, and so on affect the function of the brain. So you can pick up a peripheral signature that is reflective of brain activity. And there are similar gene expression changes in brain cells, in blood cells, due to having similar genes or promoters of genes and responding with gene expression in similar ways to the shared internal milieu or external environmental changes like medications and so on. So it's possible to find biomarkers, but it's like needle in the haystack. So we had to have a very careful approach to find true signal that's reproducible and reflective of what's happening in the brain. Next slide, please. If you found them, however, they are very useful. So they can be used for diagnostics, they can be used to guide treatments or therapeutics, and also to discover new drugs. And last but not least, they can be used to understand the biology of the disorder. Next slide, please. So these are the 4 steps that I mentioned for biomarker discovery. We wanted to have sort of a comprehensive gold standard approach to discover biomarkers with these 4 steps: discovery, prioritization, validation, and testing. Our discovery is done, as opposed to using large cohorts of so-called patients and so-called controls, we focused on people who have a disorder and follow them longitudinally over many years to see what changes when they are sick and when they're not. It's a sort of a within subject design. It's very powerful, very hard to do. So when we were starting this, at about the same time, Mike Snyder at Stanford published a beautiful, independently did a similar study uh, with a within subject design. Somebody followed over a year with an N of 1. Beautiful paper. He published it in Cell. He was the N of 1. He did it on himself. He took samples of himself over a year and did the multi-omic studies and showed that it's very informative to catch and predict future health outcomes. Our discovery cohorts have multiple mini Mike Snyders inside them, as people who we follow longitudinally over time. And then rely, once you have some candidate biomarkers, rely on multiple lines of evidence for cross-validating them, for then assess reproducibility, test them in independent cohorts, and so on. So next slide, please. So nowadays, some of those tests that we've developed and published are now available to doctors through the startup Mindex Sciences, and people always ask how accurate these tests are. Well, they're more accurate than nothing, right? Right now in clinical practice, we use nothing, routine clinical practice. But, conservatively speaking, most people that we've tested with these different tests, they're at over 70%. So in 3 out of 4 people, you can classify them correctly, and that's in all comers. Our digital tests are even more precise than the molecular tests, but they're not always objective. You have to rely on what the patient is telling you, and the molecular tests, when you test them in high risk groups, they are over 90% accurate, and when you combine the molecular testing and the phenomic testing, but with digital instruments, you get very good predictive accuracy that's comparable to anything else out there in medicine. So the end sort of products that are available now are an app that assesses the phenomic part and a blood test that can be provide reports on different these disease indications. And when you combine them, you can have very good predictive accuracy. So here in this slide, next slide, please, we illustrate that for suicide, which is a very tough problem in psychiatry. We lose a lot of patients to suicide in society in general. We lose a lot of patients. We lose a lot of people to this sort of needless tragedies. So over the last decade, we developed blood tests, biomarkers for suicide. Um, we published them. So sort of funny fact for those of you who are professional scientists, our first study in 2013, we were so enthused about our breakthrough that we submitted it to Nature. First, it got triaged, not even sent for review. Then we submitted it to Science. It got triaged, not sent for review. Then we published it in Molecular Psychiatry, which is a very good specialty journal, a Nature journal in our field. So after we published it, then Nature and Science each wrote like a 2-3 page news and views commentary about the paper, where they could have just sent for review in the first place. But yeah, so for those of you who are young, it does matter where you publish initially. In the long term, and for the sort of the field of science as a whole, what matters is that you do good work. A study, even if it's not published in one of these tabloid journals, if it's a good study, will still stand the test of time, get highly cited, and so on. And conversely, some of these tabloid journals have papers that don't get reproduced because people rush to publish or bias things so that they look more sensational and so on. So worry about doing good science, not necessarily about where you publish. Good science will always get cited in the long run. And our work has started to be cited a lot and written up like in New England Journal of Medicine, Journal Watch, which is something that physicians read, which is important for us because we want people to know that these things are starting to be available and that there's hope for their patients. And here on the right hand side, it shows the area under the curve. That's sort of a measure of prediction. The higher, the better. Area under the curve for receiver operating curve. So in a lot of the populations that we studied, in all individuals or in people with different psychiatric diagnosis, our suicide risk predictive score is high and highly significant. Those asterisks mean statistically significant. 1 asterisk, 2 asterisks means survived Bonferroni correction for all the tests performed. So it's possible. We did it, and now it's available. Next slide, please. We also did similar work for pain, developing a blood test for pain, which pain is something very similar to a lot of the mental health disorders. You rely on a subjective self-report by the patient on your clinical impression to assess somebody, and then there's the risk that you match them to the wrong medications or to addictive medications, and then you have the opioid epidemic, right? So we developed a blood test for pain. We identified repurposed drugs that are non-addictive, and that's been published, so you guys can look it up. And one of the main, actual utilities of our blood test, in my opinion, or the main one, is actually not just assessing somebody in an objective way, but you can match people to medications based on their biomarker profile on existing medications, so that narrows the list of what choices doctor have. They can go more targeted. And also you can use the biomarker signature to identify existing drugs used for other disorders to repurpose them for treating your indication. And we show here an example for pain, how we identified certain existing drugs that could be repurposed for pain. Some of them are vitamins, So there's always, signal from medications and nutraceuticals. So nutraceuticals are a very interesting and viable option for people to consider or add to their medication regimen. Next slide, please. We also did similar work for depression and bipolar disorder, so mood disorder test that came out a couple of years ago, and the blood test can distinguish if it's depression, just depression, or if it's manic depression, so-called bipolar disorder. It uncovered some of the biology of the disease, which is driven by circadian clocks. And here on the right hand side, we show how you can actually, on a patient, get like a score and match them to nutraceuticals, to medication. So that's sort of like a prototype of a report for a doctor. Nowadays, over at the startup side, they do it much nicer than this. This was sort of academic work. So I'd like to acknowledge, my research groups at the IU Medical School, at the VA Medical Center that contributed to these studies over the years. We have great teams over here. We work hard and have fun. And I'll, I'll I'll spend 5-10 more minutes telling you a little bit about the practical outcome of these things. So next slide, please. Slide 20. So about 4 years ago, before we did anything in terms of spin out startups and so on, there was this case that really sort of affected us as a group, and in particular sort of was a kind of an awakening for me, which was we had a patient that after finishing our suicide biomarker studies, a year later after that, we learned that she died by suicide. And we went back and looked at, when you do research, all data is aggregated in cohorts, de-identified. You don't communicate with the doctors of those people and so on. So we went back and looked at her data, and she was at, the arrow here points to her. She was an outlier. She was at the 100 percentile of the blood biomarker panel, at the 100 percentile of the suicide assessment scale that we developed. She was very high risk, but because this was done in a research setting, there was no ability to provide feedback to her doctor, say, look, this patient is very high risk, and you need to pay extra attention to her, and consider these other medications that match her profile. There was none of that loop going back. She was just a research participant. So it dawned on me that we publish all these, we do all this work, we publish studies, there are press releases and so on, and then nothing happens. People don't get to use them or get help. Every time we publish a paper, we get inundated with emails, calls from families, patients, and so on. They want to come to get tested. And I'll tell them, look, this can't just come to my academic research lab and be tested. So we decided to support IU into us doing a spin-out company. Next slide, please. And these disorders, mental health disorders, and suicide affect everybody. It doesn't matter if you're rich, poor, any characteristic. They cut across sort of socio-demographic factors, and current treatment is completely inadequate. So here in this picture is actually a Silicon Valley executive who died by suicide, and her family was supportive and invested in the startup, and supported sort of the spin out of this technology. So it doesn't matter if you're rich or poor, you can have access to the best care at Stanford or wherever, and still, you fall through the cracks with the current way of doing things. So, I mentioned the three circles. So those are the three circles that should be used and that we developed in terms of product. So the outer layer, and that's the logo of Mindex Sciences, the startup company. The outer layer, outer circle, is digital. We assess how people feel and think and their history through an app. And that's, what I do as a doctor or any other psychiatrist does with a patient. First, you see how, you do an exam on them, a mental status exam, and see how they feel and think. You get the history, so you get all that information. The app just does it in an easier, more precise, quantitative way, so you actually have numbers and graphs for everything. The middle layer is molecular. That's the blood testing that we developed, and the inside circle, the hexagon, is the pill, is depicted a tablet, right? So you go from assessing in a more precise way somebody's phenotype to lab tests to the right medication. And that's something that has occurred for many years in other areas of medicine, right? So you do an exam, you get the history, that's the outer layer, then you get labs, middle layer, and then you start them on a targeted treatment that's occurring all the time in primary care. We brought that to psychiatry with our product. So the mission of Mindex is, and we've discussed some of these, 1 in 4 people will suffer from some mental health problems in their lifetime. Pain also is a major issue, and we want to help save and improve lives through a more precise 21st century approach. So for that, we developed this platform that goes from digital tests to molecular tests to targeted therapeutics, and a pipeline. So this is sort of the Mindex Science product pipeline. The diseases that are highlighted in red, there are already blood tests that are being offered that can be ordered by physicians for their patients. We're also working, we haven't rolled out on a blood test for schizophrenia. And next slide, please, slide 25. So this is how the process looks. You start with digital screening. Everybody should have this app on their phone. It's available in Apple Store, Google, free trial. You guys can try it out. And it's like a Fitbit for the mind. You get sort of nice quantitative measures of how you feel, how you think, the life activities that you do, how they influence how you feel and think, and so on. And you can it's all confidential. It's all on the phone. The company doesn't collect any data, but you can export it and send us a nice report by email to your therapist, your doctor, coach, family member, friend, whoever you want. It's at your discretion. And when you come to an appointment, you can bring these nice graphs and say over the last 3 months, instead of trying to remember how you felt, you just show graphs for your mood, your anxiety, and so on. Extremely useful. Very simple concept, but it wasn't done before. Then we have SX Prevent, which is a digital tool that we developed for kind of like a suicide credit risk report. Then we have the blood testing, and then we have the matching to medications and monitoring or response to treatment. And all that is integrated in a dashboard for the doctors who order these tests, so they can track their patients in there, or it can be integrated in their electronic medical records. And the approach can be used all the way from prevention, primary care prevention, to treatment, all the way to treatment in specialized psychiatric settings. Next slide, please, slide 26. So the Mindex liquid biopsy blood test, out of a single tube, you can have reports for multiple disorders. It's all done with next generation sequencing now. The physician orders it, patient gets a kit, does a blood draw, and then samples come to our lab, and then 2-3 weeks later, the physician gets a report with risk scores, current state, future risk, best matches for nutraceutical, best matching for existing medications, and so on. Next slide, please. So in the end, we want to be able to provide dashboards for people to do a better job, for doctors to do a better job with their patients, for pharma companies to do a better job in their clinical trials, for organizations, HMOs, etc., payers, insurance companies to do a better job for their populations, and last but not least, for consumers to track their own health. I think the future is consumers taking more and more charge of their health and being informed about things. And lastly, by treating mental health, you can in fact improve your longevity. One of our discoveries was that some of the genes and biology that's involved in one direction in mental health disorders like depression, stress, suicide is turned on in the opposite direction in longevity. So we call that the life switch. So by treating properly mental health, you actually flip the life switch in the direction of longevity. So that's sort of an additional benefit. You go from keeping people alive and happy to all the way to living longer. So next slide. So that's the story, and I have a few minutes for questions if you guys are interested.
Yeah, thank you so much for sharing this really extensive and wonderful work that will help a lot of people. This is really wonderful. Thank you, and for doing the work.
My pleasure. My pleasure.
And my question, it was in the news very recently for postpartum depression and also psychosis. If do you think there needs to be a very specific test, or do you think the current biomarkers you have would reliably be
Yeah,
able?
yeah, yeah. Very, very, very good question. I think that's an area where um, a lot of tragedies are occurring unnecessarily. So first of all, postpartum, I think women need more support, more attention, more close follow up. In the past, in traditional societies, you were around family, friends. It takes a village, right? And nowadays, people are more isolated, spread out, and isolation for somebody who's undergoing, all sorts of mental health challenges and stress and sleep deprivation is not good. So we think that using our app is a good way for women postpartum to keep track of how they feel and think, document that, and show that to their OBGYN, to their primary care doctor, and so on, which is very important to catch things and track them, quantify them. And we also think that our blood test may be helpful. We can measure anxiety, we can measure depression, and as I said, we're working on a test for schizophrenia, which is psychosis. So we can look at all three, and those are things that occur with those hormonal changes that occur postpartum: increased anxiety, increased depression, increased psychosis, and all the way to tragedies, suicide or infanticide and things like that.
Yeah, thank you. And then I have one more question. We had the guest speaker here a while ago, and he showed the connection between pain perception and cancer um, severity and also treatment. He showed that actually a change in pain sensitivity wasn't just the outcome of cancer, but actually was also the causal part of changing the immune response to be weaker against the cancer. Do you know about that and the connection? That I think that would be also really interesting to use your um, methods and combine that with cancer.
That it was a connection between pain and
Yes.
cancer.
So
Yeah,
yeah, that.
I mean, pain. Pain is. I don't doubt there's a bidirectional connection there, and pain. Is primarily an inflammatory process, which can be the result of sort of cancer growing and lesions, but also can make the cancer spread out more easily. So they feed on each other, and certainly, our pain test could be used to assess and treat people appropriately for cancer pain, as opposed to trying all sorts of medications that might not fit the patient. Cancer pain can be extremely intense, and it's a very interesting angle, the one that you mentioned. That's if you mitigate the pain, that might help reduce or slow down the progression of cancer. And you know I'll look up that work.
Yeah, thank you. And yeah, just let me know, Dr. Shah, and two more people just joined the stage, but let me know when your time is up, so I'll stop the the talk. Thank you.
No problem. Thank you so much for your wonderful talk. I might have two questions. One of them, I just remember one of the slide, maybe it was number 15, that
Hmm.
we had a p-value, and that was
Hmm.
the differences between the male versus female. And that was just kind of, from the perspective of the quantitative.
Yeah.
That was a lot difference between the male and female.
Yes.
Somehow when we just move to the slide number 17, when you just compare the pharmacogenomics with each
Yeah.
other, I was just wondering to ask you, because for me, for example, some of the factors like a VGFA or some of the HLA that we see in blue,
Yeah.
that you demonstrate in blue, I was wondering, is there any correlation between them? And then
Yes.
I will
Yes.
ask my
No. No.
second question.
That's a great question, and it's a very interesting, area to look at and very important to actually look at gender differences. So, so three thoughts over there. So first of all, consistently we find better biomarkers, more predictive biomarkers in women than in men. So consistently, our ability to track and predict all these different mental health indications and pain that we studied is better in women. And we don't know why that is. Could be a combination of two things. So first of all, a lot of these genes are detected in immune cells, and the immune response is perhaps stronger in women. A lot of them are X chromosome based, and so on. And women have two X chromosome, and so that could be an explanation. But the other one is more pragmatic and methodological. So when we do these studies, this discovery, we have to initially bootstrap the discovery of candidate biomarkers to how people feel and think, to their self-reports on the instruments that we developed that are now in the app. And women are much better at more accurate in terms of self-reporting. I guess they're more in touch with their feelings. They don't minimize or hide them. Guys sometimes want to be macho and so on. So we think that there's, the initial discovery is better in women. Also, when we use them for predicting in independent cohorts, again, you know, um, guys may not be as accurate in terms of describing their clinical severity, whereas women are more upfront about that. So again, our biomarkers classify them better because their clinical self-reporting is more accurate. So for those two reasons, I think the tests that we have so far are work better in women. So it's Mindex Sciences is almost like should I'm joking, but should be like a women's health company. I should sort of market or target these tests more to women, and that would be super important because in general in the past women have been understudied. Also, anxiety, depression, pain are twice more prevalent in women than men, and we have better tests for women than men. So the tests, the panels that are being done can be done by gender, and that provides increased accuracy. And the female panel is always better than the male panel.
Also, by considering inflammation, because you mentioned about the cytokines and all of those
Yeah,
components, and pro-inflammatory, I was just wondering to ask you, for example, some of the condition like after surgery pain, they are
yeah,
totally different. Still, we have a systematic inflammation, but it might be different based upon the marker that you just introduced in a number 17 slide. And I was just wondering to ask you about your point of view about that.
yeah. So I mean, pain is very heterogeneous. It's not a single disorder. None of these disorders are a single disorder. So I think using sort of a stratified approach and identifying subtypes, as we were able to do with our biomarkers in some of these disorders, is very important. And also for clinical trials and drug development, that's why we think some clinical trials fail because you are um, giving a drug that to a heterogeneous population doesn't work in everybody, and the signal from the people in which it works gets drowned by the signal in the people in which it doesn't work. So stratifying from the beginning by subtypes and identifying which population best responds, and then using that for subsequent studies, and as a using that population to give the drug to is sort of the way of the future and the rational way forward. And it's being done in cancer already all the time, right? So people get stratified by their molecular markers and get drugs based on that. It's not given to everybody with breast cancer, for example, and so on.
Yes, exactly. But when we are talking about the VGF, is totally different because we can think about hormone, but in another case, that's inflammation. However, thank you so much for your answer. I'm passing the mic to the next person.
Okay.
Hello, doctor. I just wanted to ask you about measuring treatment progress with using these biomarkers. So, as an example, there's something called the Adverse Childhood Experience Score, right? If you get a score above a certain amount, the odds of your mortality is significantly greater than people who don't have that background. However, there are medications and treatments that do help ameliorate some of these symptoms, but it's very difficult to track what it's changing and how it actually affects the patient at an individual level, right? Like, what is the timeline for this patient's recovery? To what degree have they recovered? So what I'm asking is, can you use these biomarkers to track the patient's progress?
Yeah, that's the goal. So right now, that now that they are available and that physicians have started to use that, we can sort of look at yeah, follow-up studies, clinical utility studies, where you see how the use of the biomarkers not only informs practice for the doctor but improves the patient and so on. I there's not a lot of doubt in my mind that they can be used to track that in a more objective way that eliminates biases and subjectivity. People get taken more seriously when they report something, when you have the corroborative lab information supporting it. And the app as well, documenting things daily, how you feel, how you think, the life events in a quantitative way that's visible to the doctor when you come to appointments is also very important, as opposed to sort of just a global subjective impression or recall of how things were. And there are a lot of measures that we've put in the app that have to do with quality of life, life satisfaction, a lot of things that can be used in terms of tracking how well somebody's doing and how successful the treatment is. Um, so I think we're out of time. It was really great to be with you guys. I always like to sort of. I hope there were young people in the audience, because I think it's very important for them to learn about this, be involved in this, get sort of connected to the magic and the challenges of doing science, so that they can carry forward the torch. When
Yeah, thank you.
Yeah,
Uh, yeah, that's exactly why we're doing this. We got actually feedback this year that some people went back to school because they were kind of motivated to go more into science. So, yeah, thank you so much for
well,
being here, for sharing
thank,
this.
thank you,
And
Katerina. And people
yeah,
can Google our lab. Our company can send me emails if you have any additional questions. Okay.
wonderful.
Thank
Thank
you
you
very
so
much.
much. Very kind
Have
of
a
you.
nice day.
Thank you.
All right.
Bye.
Thank you. Bye bye.
Yeah, and thank you everyone for coming. If you have more questions, as Professor Niculescu said, reach out to us, write an email, and we will be, doing our best to answering questions further on. And yeah, spread the word. I think that's a very important technology development for mental health. And yeah, I hope I hear you all tomorrow again if you are interested. We have actually two events. One by Dr. Kirkland. He will be presenting his work. It's more neuroscience technology development of bioluminescence for the imaging of the brain. And he will explain why that is important and great technology. And then Rafay, he will talk about the potential of using OpenAI on Microsoft Power Platform, and he will guide people through the exact steps how to do it. I think education for free in that regard is important for people, especially around the world where people don't have access to higher education. So we'll keep recording these rooms. Feel free to join, and then we'll spread this around the world, hopefully. Okay, so thank you so much for coming. Enjoy the rest of your day, and I'll close the room now. In 3, 2, 1. Bye, everyone. Thank you.